Today we're going to talk about
growing with fishes. Growing with fishes.
Hey everybody, welcome to the Growing with Fishes podcast, episode 200 or 355. Today we are here with Kevin McKernan, who is back again, one of our favorite guests to have on the show. He's doing a lot of really awesome work in the field of testing and all kinds of other amazing stuff that we're going to talk about today. So we thought it'd be great to have him back on. So thanks a lot for joining us again, Kevin.
Thanks, Steve. Always good to join you, to catch up on what's going on in the field. I always learn something on your cast, so I never turn it down.
Well, I appreciate it. So what have you been up to lately? I don't know if you want to still be sharing this second, or if you want to.
Oh, well, yeah, sure. We'll might as well we'll dive in there. So, oh, what have we been doing lately? Lately, we've had a lot of attention on lab shopping and microbial testing, and some of this came home to roost here in Massachusetts. We had an employee at a grow pass away from an asthma attack in a trimming room, or I'm sorry, it was a pre-roll facility where there's a lot of dust. And CDC got involved, which is like shocking to me that they helicoptered in on this case when there's, an absolute massacre going on with the vaccines, but they had time to come and check on cannabis dust. And of course they got there a little bit too late after the grow that this happened to did the right thing in that they quickly put in HVAC, which was the right move, because there was a lot of complaint online that they didn't have enough PPE, but I'm sure as many people have witnessed going through the COVID pandemic, when you don't know what the pathogen is, masking your way out of it is a disaster. You really need to change your HVAC, which is what they did do. And before the CDC got there. So the CDC got there, started testing around for microbials and for endotoxins. And the endotoxin thing caught our attention because no one's looking at that right now. And there is a lot of literature on endotoxin triggering asthma. So what is endotoxin? Endotoxin is something that ends up on gram-negative bacteria, which I think is good for your audience to know about, because you have the choice of using a lot of gram-positive bacteria that don't make it as like, biopesticides. Like Bacillus is gram-positive, and you'll find a lot of Bacillus in some of these biocontrol agents, which is, I think, is the right. I think I've been in your cast before saying this, pounding on this, that these total tests that we have in the marketplace, the one that you're seeing up here is a total yeast and mold test, and they have some arbitrary limit of 10,000 CFUs per gram, and in some states this is getting bounced up to 100,000 CFUs per gram. And the reason it's bouncing all over the place is no one knows what it should be set at. There's really no clinical literature that says total, X amount of total yeast and mold kills somebody. There's a reason for that, is that all the clinical literature demands that you perform Koch's postulates, which is that you isolate an organism, a single organism, and reintroduce it to another host and show that it recapitulates the illness. And if you don't do that, then you haven't proven that the microbes cause the death. And that's impossible to do on total tests, because you're measuring, you're measuring everything. Well, you're pretending to measure everything. The actual tests people use don't measure everything, because not everything cultures. But so anyway, we've always been opposed to these types of tests. We're forced to make them and sell them because our labs all are forced to run them in certain states. But we've tried to educate every regulator that you're really doing disservice to the industry, because when you put stringent levels on these things, you start to induce pesticide use, which, yes, we test for pesticides, but there's always one that's not on the list. And I think California is starting to learn this. So you'd rather have biocontrol agents where you use harmless to human bacteria that can help push out the pathogenic species. So this is known with Aspergillus. You can flood these things, Bacillus and other things, and it will push Aspergillus out of its niche. And then you've got these things under control. The reason you can't do that in some jurisdictions is that they have these total tests which flag you for using Bacillus. And now to bring this back to Lorna's fortunate death, we need to be differentiating between gram positive and gram negatives. Gram negatives make endotoxin, and maybe that's a risk. We haven't proven that yet. It's just the CDC raised it. we then went and started measuring endotoxin on all types of cannabis, and it's everywhere at log scales higher than the DCOS limits. And it's also been on tobacco. And there's some literature in the tobacco field that really pins the inflammation in tobacco, not on nicotine, but on endotoxins. Right? So it's been overlooked, and the whole risk in smoking cigarettes is probably not related to nicotine or tobacco flower. It's probably the endotoxin that's growing on there that's creating inflammation in the lungs. So that's something that it's not going to be regulated in any states, at least not now, and probably not ever. It's just something that I think when people are deploying these things, that's something to keep in mind. If you have a choice between a gram positive or gram negative, use a gram positive. That way, the endotoxin loads are lower. Maybe they'll institute measuring the toxin itself like a mycotoxin somewhere, but that requires, right now there's some assays that run on a really cheap fluorometer that can, it's probably be the cheapest test anyone can run in a cannabis lab if they were to use these LAL assays. But I don't suspect anyone's gonna jump very very quickly on that. But anyway, Lorna's death got everybody looking at the labs. Okay, and what you have here is an honest lab in Massachusetts, and this shows total yeast and mold failure rates. On the scale there is a log scale on the Y scale, which is the colony forming units. So 10 to the fourth is 10,000 CFUs, which is the action limit here in Mass. And you can see this lab has got an even distribution above and below the line. Technically, if you were to count this, the number above the line is actually only about 14% failure rate. But that's still high.
Can you drag that window up a little bit? You just cut off the bottom a little bit. I just got moved there when we went live or something. I'm not quite sure. There we go. Now we can see it. Thank you.
All right.
Sorry.
Oh yeah, the test date's getting cut off. All right, so that's what you'd expect to see. That's kind of what we see in a lot of other states that have total yeast mold testing. But there are labs in our state that are doing this, and that's really fishy, right? Because nothing's really breaking everything seems to stop right at the limit. It's like the mold know not to break the limit. This is probably a 3% fail rate. So this lab is picking up all the business right now. And all the lab shopping's now, moving over to this lab. And the CCC here in Massachusetts is just in this turmoil of firing everybody. They've been in disarray for a couple of years, so nothing's getting policed here. And many of the good labs are suffering as a result of this. This tends to coincide with the lab also having elevated THC levels. We don't have this built into this portal, but a little bit on this portal. This is actually thanks to some of the hard work at MCR. Yasha Khan presented at CanMed on this topic, and he piqued our interest, and we asked him for the data to confirm it. He told us just go to the state and get it yourself. So he's not in the picture, which we did. We went to the Massachusetts state and got the most recent batch of this data, and it confirmed what Yasha presented at CanMed. But they've since now put out many more states. We don't have them all in here yet. We're slowly loading them in, but I think eventually this will probably be, I think it's about 15 to 17 states that he's gotten. He's asked everyone that has a program, but a lot of the FOIAs get denied. And that's an area that I think that we can do better as a cannabis community, is really demand transparency on this so that we can see this going on, and we can learn from it as well. Like, this is really the first report card many of these labs are ever getting on their performance, right? So I have a little sympathy for some labs, because like this has been running for years, but none of the labs really know how well they perform next to others. So this might be the first time these labs are learning that they're way off the charts, either high or low on their pass rates, and that maybe something needs to be tweaked in their lab. There's some of this going on in Michigan as well. Michigan has all types of lawsuits running over this stuff, where labs are suing the regulators, and there's a lot of lawsuits now breaking out in California over this problem. So it's gotten to be a bit of a mess, and there's not a clear-cut villain here. I know that there was a big to-do in Oregon, or Oregon. I can show you Oregon. Oregon actually had one lab that was actually correct, calibrated, and doing the most volume, and they only had about a 4% fail rate. I think I can go there on Aspergillus. But there were a bunch of other labs that had very low volume, and This doesn't need a log scale. This needs to be, I'm sorry, I'm on the wrong graph mode here. Where are we doing? Oh, pooled is, I think is what we're doing. Aspergillus here. What happened to, Oregon is this type of data, sorry. A couple different graph types in here. And if you do all Aspergillus, okay, so one thing you'll notice here, this is Oregon's Aspergillus diarrhea. This is what created the lawsuit that got this testing removed in Oregon. And, the growers, as much as we protested that, obviously we're conflicted. We make these tests, but we think they're the only test that actually is like measures a real potential risk in the cannabis field. Like if you're not going to test for Aspergillus, why test for anything? Nothing else in the plants kill people. So this is the only one that I think we can back in court. Whereas I think if growers were to take some regulators to court over total yeast and mold, they'd probably win because no one can defend those things clinically. There's any clinical literature saying 11,000 CFUs kills people. Whereas Aspergillus, you can find that literature. And the CDC even ran a study that showed that there's a 3.5-fold increase in fungal infections in cannabis users. They had to pull this data from insurance ICD-10 codes. And top of that list was Aspergillus and Mucor were the two ones that were giving them the most fungal infections. All right, so there's actually a case to be made for this. This of any safety test out there, I think this is the one that's got the strongest legs in court. But what actually happened in Oregon is the lab doing most of the testing, which you'll see is this one here, did around 2,500, 2,600 of the tests, had about a 2% fail rate. That's actually in line with Nevada and Michigan. Michigan has about a 3.88% fail rate. Nevada has like a 2.5 or something fail rate for Aspergillus, and that's over years. This is a really small window. But what happened here is a couple labs were booting up that had even like 60% fail rates. And if any grows got hit by this, they got screwed. And I suspect these are the ones that protested and put forward the lawsuit. But you have to notice that this was a very low volume lab. They had maybe barely 100 tests done on their belt. If you open this up to even some of the smaller labs, like a minimum test of 1, you can see all of these labs that failed 100%. That's not real. That's a lab not having gotten booted up correctly, and they're not in line with what we're seeing in other states. And if the regulators, I think, had this data at hand, they've been able to be like, okay, let's only look at the labs that have actually done this more than like 200 times or whatever, and it would have narrowed it down to 3 labs that have a 1%, a 2%, a 10% failure rate. And it would have changed their thinking on, okay, maybe we have some outlier labs here that are really screwing up the market, and we have to give them more burn-in time to sort this out. But this created a huge stink there, a lawsuit. The regulators folded pretty quickly on it and didn't really contest it in court and just removed the regs. And there's now concern that there could be aspergillosis happening in Oregon because no one's really measuring this. There's no VAERS system for these types of cannabis injuries. In fact, the CDC doesn't monitor aspergillosis. They can only pull that data out of insurance codes. But the insurance data on it is pretty rich in that they know there's about 15,000 cases a year, and at least that was measured back in, I think, 2009 is when that number comes from. So it could be higher now. It was definitely a problem during COVID. A lot of the COVID deaths are actually Aspergillus. Not that they were necessarily smoking weed, but there is a problem with Aspergillus in hospital air systems. They tend to have like sick building syndrome is something that you can sometimes find Aspergillus in. So but the issue with them is that they're $100,000 to treat, and if you don't treat it, it's fatal. So it's a really expensive treatment when people get aspergillosis, and so the bill in 2009 annually was like 1.2 to 1.5 billion a year. And the case we've been trying to make to people is that, look, this is a $10 to $100 test. Like it's 10 coming out of our door, and the labs mark it up with their labor and their work, and it's probably rarely above $40, but let's just say 100 for easy math. This is usually getting applied on 1 gram being tested out of 50 pounds. So it's, pick your number for cost per pound, but the spot price I looked up today was 900. That seemed a little high. I think it's probably more like 500, right? In your neck of the woods. But, either way, you're talking about $15,000 to $45,000. You're adding in $100 of cost to try to stem a $1.2 billion insurance bill. Now, the 15,000 Aspergillus cases in that we have a year are not all cannabis users. That they estimate a third of them are, but there's some numbers on like cannabis use in the United States, and it varies on state, but it's like I think the average was like 24% or something. But Vermont and Oregon are really high; they're like 31% cannabis use rates. So that's a person who's used it at least once a year. So um, so anyway, this tool we've built to try to get all this data public so that people can comb through the different states and begin to see trends like this to see are we dealing with an anomaly that we're overreacting to, or are these labs like in line with historical numbers that we see in other states? And are there any oddities like this lab shopping stuff that's going on that could be induced by total yeast and mold testing? I don't think there's a lot of evidence for lab shopping on Aspergillus right now in Michigan. If you look at Michigan, they have a lot of labs in Michigan. All right. That is Aspergillus testing, where the state is at 3.88%. And yeah, there's a couple in here that have run very few numbers of tests and their rates are a little bit higher, but the really high volume labs, I'll take that so it's not normalized, you'll see, don't have very high fail rates. So some of these big ones like 48, 42, 28, and 26, their fail rates are in line with the 3% or 4% state average. So I don't think this is probably driving too much lab shopping. However, total yeast and mold is another beast. And when you start getting 15% failure rates, there's going to be a motivation for people to move to a lab that has a 2% failure rate. And so you can see this guy here at 41% is going to struggle. They're way out of spec compared to everybody else. Now, keep in mind, in Michigan, there's really only 25 labs, but each lab has two numbers in here for their medical program and their rec program, and the medical program has stricter regs. So you're probably seeing some of these high ones on the medical program. They're at 10,000 CFUs. The rec program's at 100,000 CFUs. So these might just be medical versions of the lab. And if you take those out, it's probably going to, average it around, 10% or something like that. But that's enough failure to probably make people move. We don't see any evidence of E. coli and Salmonella doing this. Their failure rates are too low. So I don't think those are driving any lab shopping. But there's definitely evidence of it in Massachusetts of lab shopping. There's some evidence we've seen in Rhode Island, in Maryland, that the labs that have the lower fail rates for these tests end up building market share and the other ones end up dropping. Note to self, this one lab that is doing something very bizarre in Massachusetts is in 10 other states. So there may be something with that platform that they're using. We're still trying to sort that out. What type of lab was that? I think that was in this thing here. So we don't have Aspergillus here. We only have TYM and mass here. Sorry, this thing sometimes takes some time to refresh, but
oh, this is super fascinating. It's interesting to see everything laid out nice and cleanly like this.
we need transparency. We're not going to get anywhere with. I mean, one of the problems I have with what's going on in California is there isn't any transparency in what the DCC is doing. All right, so yeah, I'm concerned there's lab shopping there, and that's a problem we need to solve. The way the DCC is going about solving it is they're using an Aspergillus test that no one else can get their hands on. It's a thermo test, or at least their methods aren't even clear. We're speculating it's a thermo test because that's what we know they used to use. So I'll say that I'm speculating there, but they're still using that thermo test. No one else can order it. The company doesn't have it online for everyone to order, and no one has not been through AOAC. So they're going to throw out all of these fails in California or these recalls based on a test that no one can validate. And then it's going to be whack-a-mole on business and people's businesses. And it sucks because yes, there is some lab shopping probably going on in some out of spec labs, but the way they're going about policing it is going to invite lawsuits like they have in Michigan. Michigan got sued for this very reason, is that Veritas challenged them on what they were using as a validated method to say that they were out of spec. And they've got a valid point there. That Michigan doesn't have their own testing lab, so they relied on the Veritas competitors to claim that they were out of spec. And I don't know if they did that in a perfectly blinded manner, and if it did enough replicas. That's all what all the whole court case is contesting, is that What are the grounds upon which the state is determining what a recall is? And if they're not really clear and transparent about that, they don't do it with high replication, and they probably really, when it comes to that, when lawsuits start flying, you should really get sequencing involved. And if you truly believe they missed something, prove it's not off target, because some of these PCR tests will amplify Aspergillus that's off the 4 lists. Like we already know, there's a Bio-Rad kit out there that the CDPH from Colorado put out a poster on, showing that it doesn't just hit Aspergillus nigrotarius flavus fugatus, it also hits this thing called tamari, which is not a pathogenic Aspergillus, but that's in cannabis. We find it when we sequence things. So if the state's using that, they really need to sequence to prove it's not tamari before they do a recall. Otherwise, the lab's going to have a valid case suing them, saying I mean, the case of Michigan, like they recalled 60% of the cannabis in the state. It's like you're asking for a lawsuit if you do that. That's just there's so much at stake that the growers are going to sue the lab for getting it wrong, so the lab has to sue the regulators, and to stay alive. So I think that's where that whole thing snowballed from is. If the state's going to regulate this, they have to prove that their test is as good as everyone else's, and they probably have to run all the tests, all the vendors' tests to see, okay, does it fail on 3 out of 4 of them? Then okay, maybe we can draw a consensus from that. But if it fails on just one of them, it could be one of these artifacts. And if they pick a single vendor's test, they invite all types of regulatory capture, because let's say they pick Bio-Rad's test. Well, then all the labs are going to want to switch to Bio-Rad, and then there's only one test serving the entire market, and that creates a huge motivation for Bio-Rad to go and buy equipment and drop in free equipment in all the regulators' hands, so that all the state labs run one thing. I think the market's a little too early for that. Like we don't know that one test is perfect, and the way we're going to figure it out is by putting all this data public and seeing like what are the fail rates for all these different tests and different vendors. Like is MGC too high? Are we too low? Like we don't know these things. We know we got through AOAC, but we haven't done a bake off against all the other tests to know who's right and who's wrong. And it might be a little premature for them to be doing product recalls when they don't have all that sorted out. So anyway, it's a complicated space, but I think the right thing to do is what MCR is leading here with is put the data public, get 100 data jockeys on the internet that care about this stuff, and start putting it and start charting it in different ways to find trends, and see if there's if there evidence of lab shopping would be, okay, you have the lowest fail rate in the state and you're sucking up all the volume now, and other ones with higher fail rates are going down. There should be some dialogue with the regulators that, okay, who's right and who's wrong here is one I mean, this platform, to me, when I look at this, I have issues with the other lab in that maybe they're overcalling, but at least their data is normally distributed, right? Like this looks like fake data. It shouldn't have a cutoff there. This looks like more normally distributed data. However, I think I know the platform this lab's running, and we've seen this thing falsely call a total yeast and mold hit on Pseudomonas. So maybe it has a slightly elevated number, but it's not manipulated, right? It's at least honest. Whereas this one it seems manipulated. And if you dig out the platform they're using, it looks like they have an AOAC PTM report that shows they have a hard time calling things over 13,000 CFUs. Like there's some type of saturation or PCR inhibition that's occurring in samples that are really loaded. And so the platform has a difficult time calling above 13,000. That's like right in their AOAC report. So it could be the platform, or it could be the lab is bending numbers that we don't know the answer yet, as to how this type of stuff is being, what the source of this is. But I think just getting the data out there for him to look at is going to be, it's going to be very helpful. And for another topic on this, like pesticides, that's another one that was brought to our attention, which people were worried that, okay, when you put Aspergillus testing in place, you're just going to create pesticide failures. Well, we haven't seen that, at least in the Oregon data, and in see if I have this right here. That's state to state. I think we want. Sorry, there's a couple different graph modes here that I'm struggling to remember which one does which. Oregon, I think, has Aspergillus species, or is it? 100 data points. Sorry, this one lab doesn't have 100 data points. So intervals. So they only ran this for, I think, 4 months before they stopped it. But let's see if there's a few more of these that come through. Put them down here. That's maybe the main lab there for Flavus, at least. See if that comes up with pool. That should have all of them. Sorry, I don't know why it's doing that to me.
No worries.
I'm just so
learning a lot.
Oh, I'm on the wrong. It switched me to the wrong lab again. Oh, damn it.
Yeah, when you change the test type, it changes the lab settings.
Okay, so you can. I have this on my Substack. People can take a look at. But what we basically did is charted the testing frequency in Aspergillus and the fail rate. Initially, it started with a high fail rate because I don't think the lab, when they're booting up, they probably didn't know what they're doing, and then it came in line with all the other states, even as the volume grew. But you can see the pesticide fail rates don't move at all during this time frame. So at least in Oregon, we didn't see pesticide use go up with Aspergillus testing. I think if they put in total yeast and mold, it may be a different story, because total yeast and mold has a much higher fail rate and a much bigger economic loss involved with it. And it's probably a harder nut to crack in terms of how do you lower those numbers without remediation. Whereas Aspergillus is a very particular species. There's literature out there on like what other species push it out. So you can, when you're doing Aspergillus testing, you can use beneficial microbes because they don't trigger a TAC test. So you have at your fingertips a lot of the biocontrol agents that you don't have when you do total yeast and mold testing, and because usually total yeast and mold testing always gets implemented with TAC testing. You get total bacteria counting as well. So if you want to lower your TYMs, you can't bring in bacteria because you'll get screwed on the TAC counts. So that's one story in the pesticide front. The other story, there's an LA Times study that's come out. What's her name? Paige St. John, I think is the journalist there, which is an interesting study, but it's mostly on pens. And as we all know, the extraction process enriches pesticides, so there's a higher fail rate in that study. But it doesn't yet have a control. It doesn't have the black market, right? So they're doing that now, but you have to wait for that to come out to really make good sense out of that data. But there's a study of flowers in Canada, which is the other pesticide study worth looking at. They looked at 327 pesticides. That's the largest pesticide panel I've seen published yet. And they did look at the black market, which had a 92% positivity rate, which is crazy. Because prior to legalization, the black market only had a 30% positivity rate for pesticides. So once they legalized, the legalized market went down to 6% positivity, and the black market went to 92. So the black market became the dumping ground for the stuff that doesn't pass. I guess no surprise to anyone here. But some of the concentrations of the pesticides were like 1,000-fold higher in the black market. So there is some evidence there that, okay, when you put in flower testing up there, the legal market gets cleaner, the black market becomes a dumping ground, and people need to be aware of that. But they didn't find a whole lot of off pesticide use. They only found 2 pesticides that were off the panel that the Canada demanded you test for. I think it's 100 up there. I could have that wrong, but there was one in each bucket, one in the black market, one in the legal market that was off the list. And so the people aren't, at least in that study, they're not quickly jumping to these off-label things. The LA Times studies looking at pens, they found mostly insecticides, like things associated with these bug bombs, like you were bringing up that Hass Church episode, and wasn't Josh, who was it? Alec Dixon was going through the fact that you can see signatures of bug bombs because there's usually three pesticides in there, and they're insecticides, they're not fungicides per se. But they did have some myclobutanol in there still. There was like 11% failure rate for myclobutanol, which is awful. That stuff is hydrogen cyanide in pens, shouldn't be there. But that is lower than when Josh Werzer presented on that in 2016 at CanMed. He got up and showed the pesticide rates then when there wasn't any testing involved. So his sensitivity of the assay was lower, and people weren't being flagged for this stuff. So the use rate was like 40% back then. they're getting 40% fails for just myclobutanol. And I think it went up to 50% when you include everything else. So he has a good presentation on that on CanMed from 2016 on the pesticide failure rate. And it was actually cleanest in the Emerald Cup, which was exciting to see. So, the people bringing flowers to the Emerald Cup are the cleanest we got out there, which says something about perhaps the way we're Bud Lighting the industry.
Sorry about that. Yeah, to yell
Yeah.
at
So
my dog
anyway,
for a second.
if you're a researcher and you're interested in this data, let us know. It's not totally public yet because we're still adding in more states and kind of debugging some of these things, and making sure we didn't screw anything up because every state has a different format for this data. So importing it has been very manual and has to be curated. Like they all spell E. coli different or stack, or some call it total yeast and mold, some call it TYMC, and so importing the data in the proper field needs to be double checked, and so some of that is still under scrutiny here. But we have opened up to a few researchers who are helping us point out those problems, and then eventually we'll hopefully get it public for everyone to play with. But I think the answer to this lab shopping thing is data transparency. So all the labs can see if they're in or out of spec with other labs. All the regulators can see it. All the consumers can see it. And it doesn't require a PhD in biostatistics to comb through it and generate reports in R. If we can just solve the data display problem for people, we'll get more eyeballs on this, and it may help educate the regulators and the field on how to move forward here in a rational way.
This is important too, as a lot of new countries just starting to implement regulatory structures. Thailand's one that comes to mind. And some of the others that are, in the baby steps phase of getting into the swing of things. This is something that absolutely should be brought to their attention as well, to places even outside the U.S.
Yeah, it is it's a very, very helpful tool. I think it'll also help them guide. Some of the states now, what they're doing, I think New York did a little this, is they're raising the TYM limits to 100,000 for rec and bringing in species specific testing. We've tried to tell them just get rid of the damn test because it creates so many lab fights and creates lab shopping. But I think they're trying to balance the complaints of consumers versus the grows. The grows probably don't want it at all, and the consumers are screaming for it. But so they raise the limits and then put in Aspergillus testing. I think New York did it a little differently. They put the TYMs at 100,000, but they're not a pass-fail number. They're just for monitoring the market. So the grows don't get killed for it, but they can at least see if their numbers are going up or down over time. What's somewhat interesting in this data here, I think this is the Aspergillus in a couple labs in Michigan, and you can begin to see some seasonality here, where September, bang, you get a spike in Aspergillus in September. And this lines up with what I've heard from Zamir too. Zamir Punja says he gets botrytis indoor in Canada. He gets a much higher botrytis and other fungal spikes in September, August, September, October. And that stuff's probably showing up in the labs here in September if they're getting it in August. But it's important not I don't know what the seasonality is in Oregon, but what if they picked one of these humps when they started measuring this for 4 months, right? They may have been in a window of seasonal outbreak on this thing, and then bang, they come here and okay, there's the time to get rid of this. But the rest of the season is quite low, and that can also educate the growers that, hey, in those time frames when the dew points or relative humidity, whatever changes in the glass house, that maybe those are the times to do beneficial micro flushes or whatever your precaution is to try and lower the counts. You may not have to be doing it year round. You might just need to do it when the season's here.
Oh, that makes a ton of sense. It also makes sense that it spikes in the fall between leaves breaking down and the grass laying down and all that kind of stuff for the fall, and people cutting hay and all that stuff. It makes a lot of sense.
Yeah, the dew point changes, and I'm sure there's a moisture issue there. Sun's probably not drying these things out as much, but there's so much data in here that's going to be helpful for the field. Now the other thing that we can link this to, which has been helpful. I wonder if I can share that screen, if I can find that. No, that's not it. let me see. I may have to. Oh no, that's not it. Let me kill these windows. What window are you seeing? Zoom. Uh, there we go. All right, that's the testing window. So, um, here, let me stop the share. I'll see if I can find a pull up into the window. But the other data set we have here that can help inform this is we've done about, oh, Canopy will have it. Yeah, that's what we want to share. Okay, I think you've all seen this before, but so a lot of the folks who are now sequencing genomes with us are now doing whole genome shotgun because the sequencing price we've gotten down to the point where it makes more sense to do the whole genome than to do like the targeted genes. And when you do this, you can get a nice see, Zamir's got a bunch of genomes in here. Oops, where'd that go? What are you seeing right now on the screen? Do I have your do you have Canopy up there? Yeah.
Yeah, it says Canopedia. You're just searching for Zamir's.
There you go. Okay, so he's got he's been doing a lot of work tracking the microbiomes through different tissues of the plant. And so when you sequence the entire cannabis plant, you get all the DNA that's there. So you get the bacteria, you get the fungi, you get the viruses. We don't get the RNA viruses, we get the DNA viruses, but still interesting plots there. And you can go in here and get these beautiful classifications of all the fungi that are present. And so that's the fungal one. And then, of course, if you want to look at some of the bacteria, I mean, first off, if you look at the entire microbiome, you'll see in this particular tissue he's in, like 98% of it is cannabis DNA. So this is helpful to know how much microbiome you've got present, because you can see it's only 2% microbiome. And when you start going into roots, this thing goes down to like 80%, like 20% becomes microbiome. So there's a lot more microbiome present in the roots than there is in the flowers. So no surprise there. And then we split it out into bacteria and viral, so you can get a blue. And so you can see this person's got some Bacillus cereus in here, which is great. All these bacilli are great biocontrol agents. And these ones are gram positive. These ones over here are gram negative, the Pseudomonas ones. So you don't want these. You want more of these if you can pull it off. And then you can occasionally dig through here and find some kind of bizarre viruses that are present that we're still trying to organize, because some of these viruses we're quite surprised that it could be the closest thing in the database is something that you'd never find in cannabis. It's just hinting at something that's close. I think the issue we have is the databases in NCBI don't have a lot of cannabis viruses in them, so they tend to associate the reads with whatever's closest. So these may not be. Don't go chasing after these viruses. I don't think they exist. I think this is just a shadow at the moment until we get the more cannabis viruses in the database. But let's see if we can find you a root sample here. You'll see a completely different picture. Just got to find ones that say root.
So there's RT in some of the names.
Yeah. Oh, there we go. This one's roots for sure. Yeah, those could be roots. Yeah. So you get here. Wait a minute. What did I click on? That's a fungal. Yeah. Total microbiomes up here. Yeah. You get here and you get more like 11% of its microbiome now instead of 2%. So a lot more activity, a lot more to look at, and a lot more information rich in here as to, there's a lot of penicilliums and aspergillus. So this Terrius ones could be a concern. And this is the next question we get is how much of a concern on this chart should we be worried about in terms of like, will we fail a test? And we're starting to calibrate that now, because it's from our estimates on where the PCR tests fall is you probably need about 500 to 1,000 reads per million reads for you to have enough Aspergillus, Terrius, Niger, Flavus fumigatus to actually trigger a PCR test. It won't trigger on some of these very, very low amounts because they're just, in this case, it's let me see, I think up in the corner here, I can't see what it's pointing to, but where to go just to Aspergillus Terrius, it would tell you there's, this is 4,000 reads. And we have about 1 million reads there, right? And of fungi. Yeah, so this one might actually trigger a fail on a test based on the level it has there. But We're still in the process of calibrating that, so we can teach people, like if we sequence the genome of their plant, that okay, you have is there any amount here to worry about in terms of triggering any of the tests that are out there. But this helps us get a picture of, okay, what are all the things we actually find on cannabis? Because right now they're running off making regulations for shit we never find in cannabis, right? The food industry's here. And so the food industry is going to like jam their regulations up our ass that they want to build testing against. And so they're putting listeria in. And we can't find listeria in any of this data. Like we've got 1,000 microbiomes in here. There's no listeria. All right. So obviously it's not something that grows in cannabis. Maybe you get it contaminated from a kitchen that's handling milk or something. I mean, that's a different issue altogether. So maybe you got to do it on edibles. But there's no point in testing that on flower. It's just we have no evidence of ever being there. It's just a revenue stream for the food industry, and sadly, a lot of the food industry does have a lot of sway and influence at AOAC, because the AOAC predominantly was an agricultural certification body. So the food industry has been, running that thing. And as a result, they kind of get what they want, and they've managed to push listeria into the field. If we did this more intelligently, I'd say if you're really worried about another pathogen, I'd go after one of these, penicilliums or something that are known to make a mycotoxin, or something that's frequently there and known to hurt people, but not something that just happens to have a test off the shelf you can suddenly slap onto our industry. So we're trying to get this data in front of regulators and growers and testing labs. They have a basis upon which to make sound regulations that aren't just based on, hey, let's just superimpose the food industry onto cannabis and force them to turn into a Monsanto like ag industry where everything's upside down, inverted, and backwards.
Yeah, no, I know I'm super excited to dig through some of the microbiome data and really kind of see you know what patterns and trends we're seeing in the data.
Yeah, this would be, I think, very helpful for doing just designing beneficial microbes. Because you could sequence them before, apply your mixture, look at it afterwards, and be like, okay, we pushed out the bad ones, and we've replaced them with non-pathogenic ones, and we didn't have to use pesticides. And that would be helpful to show many regulators that are putting in these total tests that like you are outlawing accidentally, you're outlawing the greenest technology we have to clean up cannabis by putting in these TAC and total yeast and mold tests. And if we would just step back a little bit here, not realize that if you do that TAC and total yeast and mold thing, you were creating a cat and mouse game on the pesticide front. Like people are going to be always induced or enticed to use a pesticide that's not on the list in that environment, and that's going to be like synthetic cannabinoids. You're going to be chasing that demon for years. So I think the right solution here is to use biology in your favor as opposed to using a lot of these pesticides. I am particularly annoyed at the whole pesticide thing just because I deal with like Alex Berenson on Twitter all the time, and this guy's running around yelling that, oh, you're creating psychosis and violence and all these things, and he points to these studies where they looked at marijuana. They don't even chemotype the thing. They have no clue how much mold or pesticides or mycotoxins are on the thing. And there's all this literature on these pesticides having neurological activity and causing psychosis. So he's creating a whole straw man that cannabis creates violence now. That's his go-to. Buy my book. I'll show you why your kids are going to get violent from cannabis. And he's on team prohibition now. It's sad to see that they can leverage a lot of the NIDA data that uses horribly QC'd cannabis. They don't even know if it's CBD or THC, and they should know that CBD is in trials for reducing psychosis. So if you don't get that right, you really can't make any claims in the schizophrenia paper. They do it anyway, but we've got no understanding of whether that's really cannabinoids or whether it's pesticides right now.
That's something too. I think a lot of the flavors that people kind of miss from back in the day were actually just pesticides. I had the whole conversation with somebody the other day that
Oh God, yeah, flavors.
some
I
of the
haven't thought about
different
that.
peelings and stuff that you got off of smoking some of that stuff wasn't from the weed.
Yeah. Oh God, what a nightmare.
Um, you had since we're still on the topic of like data cool stuff, this is really amazing with mapping out the microbiome. It's certainly something that a lot of people have kind of wanted to see, especially the more of the organic soil people and aquaponics people and some of the other people doing more microbial heavy methods. You also had a really cool thing the other day on sulfur compounds and comparing those and talking about
Oh
how those
yeah.
are really something that's kind of, a blind spot towards a lot of people's breeding programs and stuff because it's not quantified, in a lot of these other tests. I don't know if you
Yeah,
want
and
to touch on that.
that's not our work. That's the great work that was done down at Abstracts Labs.
Oh, okay.
They pioneered all that, and we're collaborating with them on another project right now that is soon to publish, um, related to some people trying to graft cannabis and hops and what types of weird genetics come out of that. But that's to yet to be published. But in that process, we got to learn a little bit about what they're measuring. And yeah, those sulfur compounds are like parts per million, parts per 100,000. They're really really low and have a huge impact on aroma and flavor and gassy smells that come out of these things. They're these sulfur compounds, and it looks like some enzyme in the plant. We haven't isolated this and know what it is, but the compounds they're finding look like the precursors to terpenes that are just have like an oxygen replaced with the sulfur group. So there's probably some enzyme that's doing that. Or someone brought up maybe some photochemistry. It could be that too. But I wouldn't be surprised if some of these plants have an enzyme that just thiolates one of these groups, one of these IPP groups that are in the process, the building blocks of terpenes, and makes a very small amount of this with sulfur. And then it's so damn potent at parts per million that it totally overpowers any of the terpenes. From a human nose standpoint, our noses are really sensitive to sulfur compounds. They happen to be emitted by like dead flesh, like cadaverine is one of them, and so the sulfur, our nose is really sensitive to like, oh, maybe that's something dead and infected. Stay away. So for whatever reason, the GPCRs in our nose and our olfactory receptors are highly tuned to these style compounds.
Well,
And
it makes me wonder too if it's not a microbiome issue too. So like if you use
yeah, it's a good
actinic
point.
if you use actinovate, for instance, in aquaponics, it'll actually make your fish taste off. It has a secondary compound that turns into a terpene that builds up in the fish tissue and makes them taste kind of earthy and like
Oh, really?
kind of
Yeah.
spoiled. Yeah. So you
Yeah,
actually can't use actinovate in aquaculture.
we're gonna have to look at that because it is a I don't think it's been addressed yet. Like they're definitely being synthesized. Abstracts can show you that, but whether there's some sort of secondary conversion route that's occurring due to the microbiomes that are there is a very good question. I mean, they are seeing that some of these things correlate with certain strains. So, like, some of the Jack Herreras and some of the OGs that are gassier, I think they consistently are seeing them. So that does lead me to think it might be within the plant, unless those microbiomes all happen to be similar as well. I think that's something that we probably should tease out with them. We should probably get a couple of the plants that they think it's holding genetically and just see like, okay, if they have different microbiomes, then we can rule that out because even in spite of those different microbiomes, it's still giving them these compounds. But someone else on that hash church brought up the fact that some of this photochemistry happens with Heineken beer, where some like green bottle versus brown bottle, they get different photochemistry that goes on that creates a skunkier smell in one of them, and that we can't rule out photochemistry for it all. But it's a whole new arena, I think, when it comes to flavors. We've been so in aroma, we've been so terpene focused that we really have to come to figure out what that pathway is and why does it differ in the strains that tend to be gassier. Like, what is it about those genomes that might predict a gassy outcome? I mean, they're also volatile as well, so I think there is going to be a little of this. We've got to be careful on like measuring it really early when you're cutting these things, and making sure it's not trying to do this after stuff's been on the shelf for 6 months. Who knows how much of it's outgassed and whether you can pick it up. So there's there's a temporal side of this we have to pay attention to in going after those studies. But yeah, they're doing great work. I think it's TJ and Ian Oswald are over there that are really pioneering the detection methods for those things, and it will be great to have those to have that more widespread, so we can start to figure this out.
Oh, that's really awesome. I know not the last time you were on the show, but the time before you were working on a bunch of work with hop latent viroid. Do you have any updates on that? And then are you doing any other type of disease testing as well and on other stuff that you guys are working on?
Yeah, we are. We're doing a lot of stuff on hop latent. Let me see if I can figure out where we are on that. That is what was the last convo we had about that? Just so I don't repeat myself
Sure.
here.
I think you had just come out with that mapping tool for mapping out that had like all the different diagrams, the different
Okay. All right. Yeah.
latents.
That's all right. So that is live, and so yeah, we have done some more work. Let me just find that post. So after we did that, we got a collection of all of the different sequences that are around the world. That's on a Viropedia page that I can point people toward that has a mapping of, I think there's 193 different viroid sequences in there. And when you go through and curate a lot of them, there's really two dominant strains of hop latent viroid that are out there. The NCBI will lead you down a rabbit hole thinking there's 50 different strains, but it turns out if you go through and curate a lot of those, there's a lot of sequencing errors in them, and we're less convinced that that diversity exists anymore. It's really dwindled down to just the two, which is interesting in of itself, because the literature suggested this was going to be a quasi-species swarm, and it'd be a lot of error prone, and we'd have all types of SNPs. But we went about sequencing samples from all over the country, and when we really take care to sequence them with multiple sequencers and rule out all the errors, this really boils down to just CAN1 and CAN2, which who are the guys at it's not Dark Horse, Dark Heart Nursery, I think, who originally reported that.
Yeah.
And so I don't think it's diverged much from there. Now, what that got us doing is saying, okay, if there's only two out there, then we should really dig into Koch's postulates, because we don't want to repeat the whole COVID madness here, where we create like a PCR scare that isn't fucking real. And let me see if this one has the study on it. That's one of them. I'm going to keep that one open. We'll go to that one in a second. Sorry, I'm just digging up this other one. But so Koch's postulates, I mentioned this earlier about how the responsible thing to do in any type of biology like this is to prove that the infectious agent is the only agent that is causing the illness. And whenever you're dealing with latent viruses or viroids, you have a kind of a duty to do this, because they can be really tricky. Just like with COVID, there was a lot of concern over, hey, did they really perform Koch's postulates, isolate the virus, and is it only the virus? There's all this comorbidity going on. Is this thing just a shadow that we're chasing? And it's really hospital protocol and most of the crap that's killing people. And there's a good argument for a lot of that. But yeah, they've isolated the virus in COVID to my satisfaction, and proven that it can replicate and given to another person. How much disease it causes another person, that gets more complicated, because depending on who you give it to, if they've got diabetes or COPD, and you start putting them on vents and giving them and stripping away antibiotics, yeah, you can hurt somebody. But the virus in and of itself, it's rarely lethal unless it's mistreated. I got really sick from it, so I'm sympathetic to it, and I thought I tried to treat it well, but now I got to find the share button for this. Oh, I lost you, man. I don't know where my oh, there. Here we are. Too many damn windows.
No worries.
Sorry about that. Here we
Same
go.
problem
I should be able to find it now. Are you like me and you have like 50 screens open at once? And
Yes, I have my different
okay,
sets of windows for different projects.
Okay, what do you see now? You got that should do it, right?
There we go. Now we're
Okay,
looking
so all right, so this is you were mentioning, this is the little Viropedia page we made where it charts the sequence of the viroids and all the homology of the cannabis genome, and this changes a little bit between CAN1 and CAN2, but it changes a lot in a lot of those other ones in NCBI, and they're usually thermal mutants. Like people try to really heat the plant up to high levels, so there'd be more mistakes in replication, and then they find a lot more variation. But I don't think that's happening actually in the field. And when people send us stuff to sequence, we've been getting this kind of distribution of hop latent viroids, CAN1 and CAN2. So they're both all over the world. So what we then did is we contacted GenScript to actually chemically synthesize the viroid as a pure circular form of RNA, which they did for us here. So now when you inject this into a plant, it's only one thing. And if the plant gets sick, you know what it is, right? In the past, what everyone was doing was grinding up sick plants, and those sick plants usually had Fusarium and Pythium and a bunch of other shit. After they get hit with hop latent viroid, their immune system goes off, and who knows who's driving that bus at that point. So we never like doing is grinding up infected plant and infecting other plants with that, because you don't know what else you're bringing along for the ride. Even if you do cuttings, maybe you bring in some Fusarium spores or something else with it. So to rule that out, what you do in plant pathology, in any pathology, is you isolate the virus as best you can. In the case of the viroids, we can actually isolate it perfectly because it's chemically synthesized. We don't have to capture some viral capsid and worry about how well our centrifugation isolated the capsids from everything else. We can tell the chemists, make us exactly this circular piece of RNA. And then we can take that and inject it at different doses. We can put it in the stuff I've been doing. I've been putting in like 700 billion copies of this thing into a plant. And once it goes into flowering, bang, it's everywhere. Starts replicating. So we have these now, and this has actually been really helpful for improving the sensitivity of our tests. There's a lot of tests out there in the market that when they try to prove their sensitivity in terms of how low they can go, they cheat in that they feed it DNA. And synthesized DNA is much easier to amplify because in these types of PCR assays, you actually have a two enzyme process that goes on. You have a reverse transcriptase assay that turns the RNA into DNA, and then you have a qPCR step that amplifies the DNA. And if you just feed the test DNA, you get these really optimistic, hey, our test is sensitive down to X number of copies of DNA. But the hardest part of the assay is turning the RNA into DNA. And so you're not really torture testing your assay truthfully if you do that. One of the reasons we had this made is we wanted to prove to the world that, look, we have sensitivity at the RNA level, and it has to be a circle when you do that, because when they're made into a circle, they're these tight little like indestructible hairpins that you keep hearing of people having a hard time killing this stuff. That's because it's a tight circular piece of RNA, and that stuff's really hard to degrade. So it's also hard to amplify. So when you feed the assay linear DNA, you get completely different results than when you feed it circular RNA. So we went through the painstaking process of optimizing our whole assay on circular RNA, and I'm pretty certain we have the most sensitive assay out there for this. So if you're going to look for really low copies of this, this is the way to do it. Anyway, now that we have this tool, what we've been looking at in the plants is where in the plant are we seeing the viroid and when? Because a lot of folks in the industry went on this bandwagon of test everything in the roots, including us. Like we were like, yeah, we find it more in the roots. What we've learned since we've been doing that is there are a lot of plants out there that keep it in the roots and never goes to canopy. And that's interesting information because it's unclear whether those plants are going to have a yield loss. They're probably still an infectious concern. You got to worry about people cutting the roots and moving it around, but it's not as much of a house on fire if your mother plants keep it in the roots, because it probably won't transfer on cloning. That's an area that we have to do a lot more work on. But having these like purified constructs has helped us do this, because there's some other things we've been able to learn about this viroid, is that when it's going north into the canopy, it tends to go into replication mode, which means the circles start making this linear stuff. And this linear stuff gets processed into more circles, and the process kind of repeats itself. So we built a tool to measure this process, which is how do we differentiate between linear and circles? If it's circles, it's latent. If it's got circles plus linear, it's actively replicating, and you got to watch out. There's an enzyme actually that will destroy linear RNA, but won't destroy circles. It's RNase R. So we use this enzyme to do qPCR before and after treatment of this, and we can measure those offsets so we can gauge, okay, this is circle plus linear, and this is Post-treatment, this is just the circles. And when we get this like CT offset, we can quantitate, okay, how much is circle versus linear, and begin to look at that throughout different tissues. I have some of this on I guess a post, but oh, there's a cool new cycler we're using too that's like $10,000, which is great. It's cheap. It's probably the cheapest cycler out there for doing this qPCR stuff, but it comes with a robot that does all this PCR setup for you, which is handy. So we went about looking at this in, where is it? A bunch of different tissues just to measure the circular to linear ratio. And where we were seeing more of it and less of it in different samples. I think we even did this in like in petioles and meristems. So if you just treat the pure circular RNA, that's kind of the ratio you get. So that's the baseline. But we are seeing some things in meristem and petiole really, really, blow up in terms of the amount of, and that's expected. Once it starts going to the canopy, it starts making this linear stuff. But I think the take-home message here is it's difficult to approach if your mom room is infected and you're worried about calling all of your moms. Don't like stop and look in the roots and in the leaves, in veg and in flower. And if you don't see it moving into the canopy and flower, you might want to breed with that thing, because that's a plant that seems to be tolerant, and it ends up, and it may not. Again, it could be an infection risk to other things, but it may not be. It may have no harm on the yield. And if you go in there and decimate everything just based on root positivity, you're going to kill everything. And that might be very counterproductive. So, we've been working with this one plant, Jamaican lion, that keeps it in the roots. Since then, Jamaican lion's not like it's a great outdoor strain. It's a 1-to-1. I think it's beta-caryophyllene dominant, but doesn't have the best flower head. It's not a production strain. It's called that, an indoor production strain. Its yield is probably maxes out at 19 or 20%, 1-to-1. So what we decided to do is see whether that trait is heritable. And so we crossed it with a mandarin cookies, which is more of a, something people want to grow. It's type 1, and selected for the type 1s that came out of that. And we're now just infecting this with this purified RNA to see if it'll keep it in the roots. And right now, the story we have on that is it is keeping it in the roots, and we couldn't even pick the damn thing up and veg in the roots. But this is what stunned me, is we had this thing in veg from I think May until August 1st, testing it, looking for this thing after we infected it. And these even came from the seeds that we made from this. We tried to also we basically got HLVD infected pollen. We infected one set of plants. The pollen became positive. We put that pollen on Jamaican lion, and we got like 2 seeds out of 40 that were positive after they sprouted. So the transmission rate was really, really low. And we took some of the ones that were clean, grew those things out into next generation. So now we've got a mandarin cookies Jamaican lion cross, and now we're on that generation infecting them with HLVD. And so far, having these things in veg for like 4 months, we can't detect it in the control, obviously, the plant we didn't infect, or in the one that was infected in the canopy or in the roots. It's only when we put it into flowering did it start to show up in the roots. And that stunned me. I was expecting to find it in the roots the whole time, but it evaded detection until we turned the lights. 2 weeks after we turned the lights, bang, it's a really high copy number in the roots. So, I mean, that's one story. That's one set of genetics. We've spoken to other people in the field. They have seen this as well. They are finding plants that keep it in the roots, and they were killing those before. And now they're starting to think differently about it, that maybe those are the plants that are going to breed our way out of this. And they're going to try to isolate those and measure the yield on those to see if there's really a yield loss. Because if there's any yield loss, then we get plants that keep it in the roots. No yield loss, we move on. But the clear cutting based on any positivity may be a little dangerous. We may lose a lot of the gene pool that way.
Yeah, no, and I remember you talking about this when it first kind of came out, about trying to find the resistant plants, and you think that's eventually going to be something you're able to see in the genome of the plants and be like, oh yeah, this one will stay in the roots and this one won't, or you think it's a mix of endophytes? Or
yeah, we're starting to look at that with with Zamir. So he sequenced all of his plants, and he had a spectrum of plants that were had high positive that had it in the canopy and the roots, and he had some that had less impact on yield. Where so hop latent viroids there, but maybe only has a 5% impact on yield, and others where it had like a 30% impact on yield. And when it had a big impact on yield, he mentioned those things were getting Pythium and Fusarium and other things. So he's got a spectrum of plants now that have a different response to this. We've sequenced those. We have to go hunting through those genomes to try and find out, what is it that's doing this. We have a few candidates that we have a hunch, but we're probably going to be wrong. The first place that we're looking at is RNase 3. That's a gene in the cannabis plant that should destroy this thing. And maybe if there's variants in there, there could be some variants in that gene that mean you're less able to deal with this. We also have a very large RNA-seq data set that a group at University of Tennessee has dug into. We gave them all that data. They had a couple grad students really tear that data apart, and they're coming up with all types of interesting findings of genes that are that are overexpressed and underexpressed in the process of Jamaican lion getting infected. A lot more signature in the roots, as you'd expect. The roots had much different, much more dramatic expression changes. And there's some interesting things there. They had some things in root development and xylem and phloem genes that were involved, but they're kind of writing that up. That's their story to tell. But they're starting to dissect the biology of, all right, when you're infected, what are the changes in gene expression in a plant that keeps this in the root? We're going to have to repeat that experiment on a plant that sends it into the canopy, because they didn't see a whole lot of change in RNA expression in the canopy of Jamaican lion, and that's to be expected. The viroid wasn't really there. So it's they did see some, so there is some signaling going on in the roots of the canopy, but it's not as dramatic as the gene expression changes in the roots. Where do we are here? Oh, this is just some of the sequencing of these things. This is Illumina sequencing, and you can see the CAN1 variant is over here. And these are some of the tubes that we're using. We're in this system now that does 10 microliter reactions. This robot I was telling you about is really slick in that it puts this oil overlay in the tubes, and it pipettes the reactions into these oil pockets. And that means you can do half reactions. You can get them down to 10 microliters. Typically, you're doing 20, so the tests are twice the price. This thing cuts the price in half, running it this way. So you can run these things very, very quickly and cheaply on this Make Moira platform. I don't know if I have a picture of that here, but there's a cool robot video on our website of this robot that's doing this. But it's about the size of a 3D printer, and it runs around and sets up these reactions into these little oil wells, and then throws it on a really tiny cycler. But the footprint of this thing is really small. You could build an HLVD detection system on a bench that could fit a laptop. I mean, that's now down to that size, that you have a laptop and then a computer with about or I should say a robot that has about the footprint of a Mac, maybe a little bit deeper than that, and then a cycler that's about the size of maybe a 4-pack of beer. And that with that footprint, some of the grows are starting to fire this up and run their checks internally. So it's already we worked with a couple grows that have used it and just wiped out their HLBD problem in a matter of a couple of months. They just did enough testing to know, culled what they had to, culled intelligently, and these CT scores are really helpful, because you'll see is some of the samples will cluster. You'll get like a bunch of plants that are out in the 30s and a bunch that are in the 20s. That's about 1,000-fold difference in viroid load. So they focus their energy on getting rid of the ones that are in the 20s, and the ones that are in the 30s, they monitor a little bit more closely, because if it stays in the 30s and flowering, odds are it's not going to have a yield issue, because it's too low. It's 1,000-fold lower than the other plants. It's probably not the plant's keeping it under control. And I think that's like an important message, because there's a lot of people just calling if they see any signal. But what you tend to see when you do a lot of this is there's two populations. There's a population that's really late and the plants are fighting it off, and there's a population that are really that have really low CTs, which means there's like 1,000-fold more viroid, and those ones you do have to call. But the late ones might be the ones you want to breed with, because they seem to be managing it. And when we have seen plants that are positive and have gone negative, like it fought it off. It was an outdoor plant. It was one of Collins. Oh, sorry about that. Didn't mean to zoom in on that. But So yeah, that's the HLVD story. Yeah, these are some of the poor suckers that we've been injecting with this stuff. That's the injection needle you use to put the. We're usually putting about 700 billion at a time. I'm dying to do a dose response curve on this. I want to do it on a different plant because this thing seems to tolerate it so well that I don't think it's a good survey. You can shower this thing in HLVD infected pollen and get very few seeds that are positive. And when you inject this, it goes into the roots, and we rarely even see it in the roots unless it's in flowering. So I want to get one that's like known to get really sick, and then start putting in a million, a billion, a trillion viroids to see, okay, what's the minimum effective dose to actually get a plant to lose yield.
Just use a cookie strain. It'll be fine.
Is it
Like
the cookies
Girl
really bad?
Scout cookies or any of that shit. All
Really?
that shit
Okay.
is super susceptible.
Yeah, we also saw this thing with Jamaican line. I don't know if this repeats in other plants, but this thing turned purple on us, and we did not see that in the control. And you can see the purple showing up in the PCR. In any area we surveyed that was purple, we'd get the internal control of the PCR, but we would not get the viroid to show up. So it wasn't impacting. The anthocyanins aren't like an inhibitor to PCR, but they're not carrying the viroid unless we got green tissue. We could find green tissue, we could get some positivity on it.
So you're saying the anthocyanin was reducing the viral load in the tissue or was present?
Um, I don't know the mechanism, but yeah, that is an immune response. So it's possible
Hmm.
that that's a canary for the immune response, that the immune response is on, and we get anthocyanin coming along for the ride. But I don't know if that's a predictor in every other plant, because maybe other plants can have the same immune response but not kick that on. It's a good question. Oh, this is it. They put a little reggae tune to this, but this is the robot I'm talking about. So you can get a size of its footprint, and you can you put your little tubes that you boil your samples in there, and this thing runs around and sets up all the PCR and these really tiny PCR reactions. These 10 microliter reactions, but it takes about 14 minutes to set up a whole plate of 48 of these tests, or I guess 46, 15 minutes. There's two controls. There's a negative and positive control, so you don't get 48. Um, but once this gets going, the cycling takes about 45 to 50 minutes. So once you get this thing setting, what it's doing here is it's taking the boil. We take biopsies of the plant and we boil them for about 10 minutes. And that liberates some of the viroid. And then we have to array those into these little tinier wells that have PCR reagents in them. This thing's setting up that PCR reaction now. It's going to distribute it to all these things. And then it will start adding one sample at a time into these other wells. Yeah, let's see, what is it? Oh yeah, so now it's going after the green samples, the lice samples, popping them in there. And when that's done, you just cap it and pop it into one of these little toasters over here. And this thing does, technically 46 samples at a time with a negative and positive control. But once that thing gets going, this is an interesting cycler. It doesn't use a Peltier. It uses a magnetic induction system, which is why it's so small. This is sped up, but you get a sense of the positive control pops up, and then all of your samples start showing up over here. And you usually want stuff that show up before 35 on our tests. Stuff after 35 is a little questionable. That I guess they were doing male female there, but you could do. We have this working on XY testing, on hop latent viroid testing, and now they've just migrated the Aspergillus testing, the E. coli, and the Salmonella testing onto the platform as well, all in a boil prep manner. So you can run all boil preps to hit some of the compliance testing as well on that thing. So you can kind of handle both ends of the problem. You can handle the plant pathogens the regulators don't care about, but you can also be looking for the things the regulators do care about in the grow itself, so that you're not having this back and forth with some of the labs. Now it's not gonna replace lab testing, because it has to be done by a third party, but you can at least screen stuff before it goes, or use it for an inline process to maybe prevent the stuff that's happening with Lorna. The thing about Lorna is no amount of lab testing really would have saved her, in that she's handling material pre-testing, so they don't know what they're grinding up, whether it's going to pass or fail. They do the compliance testing on pre-rolls that already have paper, and they include the paper in that. So by the time you get a fail from the lab on that, it's too late. The people in the pre-roll room were already exposed. If that was in fact what played a role in her asthma attack. But I mean, her cases, I think, got a lot of confounders in it, and it's hard to draw a lot of conclusions from it because it was during the pandemic. COVID could trigger asthma. The vaccines we know trigger asthma, right? So that grow was 100 employees or more, and so they probably forced vaccinated everybody there. So and again, we've been in the cannabis space long enough that we don't have people dropping from asthma like this every month or every week. So it's probably a lightning strike event that maybe occurred when there was a high load in one of these trimming environments that happened to hit an asthmatic person that maybe they had a COVID case or a vaccine recently, and it brought something to bear that we just don't see as frequently. But it's definitely getting attention at the OSHA level, and OSHA is putting out studies showing trimming facilities and that they're like 40% of the employees are exposed to endotoxin above the DCOS limits, and that there's high botrytis loads and other fungal spores in a lot of these trimming facilities. They're mostly done in hemp facilities at the moment because I think OSHA can only look at that at the moment. But there's a study that was done in Minnesota and a couple others that we've got listed where they're digging into this sort of environmental hazard and what risk does it pose to trimmers.
Oh, it's definitely good info, and it's really cool new tool that you guys got there to do a lot of testing in a short period of time. That's awesome.
Yeah, it is handy in that it's incrementally scalable. The challenge I think we've had in the past is to scale the PCR testing. You usually had to buy like $100,000 robot, and so you don't want to buy a second one until you know your lab volume is going to get that high. Now that little robot's like 30K, and the cycler's 10K, and you can daisy chain 4 cyclers to that robot. So you can kind of more incrementally scale your lab by bringing that thing in. Lower footprint, easier to distribute around your lab, and you don't have to make these huge 100K jumps and really bet on a big investment. I mean, this is the lab. I don't know how these guys do it, but I mean, they're dealing with large capital equipment expenses, and they don't have banking, right? It's really hard to get loans for this type of stuff into those labs. So it became a high priority for us. So like we got to lower the instrument cost as best we can and have smaller options for people. That way the labs can boot up without having to get a bank involved per se. If you can get some of this stuff done on a credit card, perhaps you'd be able to scale a lot more readily.
Makes
Yeah, so
sense.
that's kind of where we are on the HLVD side. We don't yet have an answer on the resistance genes, but there is more data emerging that there are plants out there that tolerate this, and that's kind of where the hops field ended up. Is they found plants that tolerate this lower alpha acid reduction, and it's not gone, but it's not as big of a financial impact on yield.
Have you guys found any other diseases or anything that you guys are tracking or
We
mapping out
did
right now, or anything else?
Yeah, in the course of all the sequencing, we did run into a cucumber mosaic virus that's in cannabis, and it was only, I think it was like 98% identical. So it's very diverged, and we're still trying to build a PCR assay for that. That's been a little bit trickier, but that did have a lot more variation actually than hop latent viroid. We're surprised by that. So that is out there. It's been seen, and the tests we have on our site right now for that are actually targeting the one from cucumber, and they don't work on the one we found in cannabis. So we're going to have to update that test because we designed it off of what we were expecting, and after we sequenced it and found it in some of these samples, it's diverged enough that we got to redesign it. So that's something we have to get our arm around. We need more RNA sequencing on different plants to find. We just, before we solidify that, we want to find it in a few more plants because we don't want to design it again and then miss because we didn't survey enough of the field for what it looks like out there. But I don't know what that does to yield. We don't have an answer on that yet. We just found it in our RNA-seq data, and it was in a plant that had HLVD. So we're like, well, we don't know which one is the driver here, so we can't really claim it's pathogenic until we isolate the damn thing and run it through Koch's postulates. That's going to be harder for that virus because it's a bigger genome, and we probably have to isolate it the old school way. So we don't have all the Koch's postulates on that ironed out.
Well, I can tell you all the mosaic plants I've seen definitely had smaller buds. Quite, some cases 30% less or more. So
Oh yeah. Yeah.
yeah,
That's the reporting. Yeah. And other plants too. Right. Yeah. So I just didn't want to speculate on what's going on with this particular cucumber one that we found in cannabis because we didn't actually get it in isolation of top
sure.
latent viroid, so we couldn't make any call on that. But um, yeah, so that's some of the things we're up to. There is if you're interested in the endotoxin work, I gave a presentation on the endotoxin stuff in at CanMed. It's all over the hemp market. I mean, the hemp market doesn't have any testing, so the stuff there tends to have really high bacterial counts, and they usually don't have to remediate. So the remediation doesn't kill the endotoxin, though. That's the thing, is it gives you this false sense of, oh, look, there's no microbes growing, but all of the shit they left behind's there. And I hope they move that direction. Like if they're going to go to remediation as opposed to cleaning this up on the front end, that the regulators start having them test pre and post radiations. The consumer knows that, hey, this thing used to be screaming hot with microbes, and they zapped it, and some of the mycotoxins and endotoxins might still be there. It's interesting because like the USP is often used to as like the Bible for what they should be doing for microbial testing. But the USP spells out that you can't remediate your way to safety. Like if you have something that's over the microbial test, remediating it beneath it should not be sold. Like you should only be, remediating things if you want to get the fungal loads or the bacterial loads lower for shelf life purposes, but it has to pass to start with. But no, and everyone's like, choosing their own adventure through that manual. So it's not happening. There's lots of people, and it's going to get worse in Delaware. I think Delaware just put in a 400 total yeast and mold limit in Delaware. Like it's the whole state's going to have to nuke their shit. Like nothing's going to pass that.
Yeah, that's crazy. I mean, would it even pass if they microwaved at all? Is that the only way to
Yeah,
really
they're going
pass
to
it?
cook it. It's just that state's destroyed. I think Iowa did something similar. Illinois is really low, so everything in Illinois is going to be cooked. Yeah, they sadly, the regulations are all over the map. And in some states, they're just, they're entirely favoring a fully irradiated process where everything's going to have to get treated some way or another in order for it to get through.
Sure. And if you guys want to find that talk, you can find it on the Medicinal Genomics YouTube channel. There's a lot of really cool, interesting talks. I've actually listened to a couple of them this week, actually, that I
Oh,
hadn't heard
we're
of.
having this in Puerto Rico next year.
Oh, cool.
So yeah, if you're gonna go to CanMed, that's the one, man. Go to Puerto Rico because that's in a beautiful resort, and it's in the Wyndham northeast corner of Puerto Rico. And I got to follow up with Mark on this, but Mark was suggesting he knows a lot of the grows down there and wants to try to organize like a grow tour, so some people from the conference can jump on a bus and go check out a grow on the island.
Yeah, I definitely know. I know a really good Korean natural farming cannabis grower down there too. I'm sure I
Yeah,
could turn you on to him.
link us up. We'd love to
Sure.
get in touch with folks in the island so we can make the most of that event. But we put it down there so it can be a little cheaper. The Marco Island was great. It was like a luxury. It's like you're going to a wedding every day because they had 3 meals a day that were glorious food on the beach, and it was like a vacation. But it did have, the price was a little pricey for everybody. We felt like we're losing some of the audience that way. So I think we got it down 25% by moving it down to Puerto Rico. And I think the flights there should be just as cheap. So
And you guys can check out all the awesome stuff that they have on their website. They got cannabis stuff, mushroom stuff. I know you launched that Psilopedia when we had talked previously on one of the other episodes.
yeah,
Tons of great
that came back to
resources.
that came into really relevant conversation with this most recent FDA recall, which is so I think it was California, but there were some like mushroom tablets that were making people sick. Did you hear about this?
No, I didn't. Oh, because
So,
they were using synthetic muscimol instead of regular muscimol. Was that it?
so maybe it was synthetic, and it wouldn't have mattered. I did see muscimol in there, and because when they were showing the symptoms of this, I'm like, that's not psilocybin. Psilocybin doesn't do that. And then I saw that there was, muscimol in there, and that's basically from Amanita. And the test that we made for Oregon tests for that. So Oregon had us make a test that speciates cubensis from wood lovers. Okay, for those who aren't familiar with wood lovers, there's a set of psilocybe that grow on wood, and under some circumstances they create some other tryptamine. We think, in fact, there's a couple really good uh, hypotheses running here for the different tryptamine it might be. There's one that's I think it's called butaphenidine or something. It's a poison frog makes it or something, but it's like one carbon group different than psilocybin, and it ends up being a paralyzing agent, acute. I mean, it wears off, right? So you get this thing known as wood lovers paralysis, and they think maybe that's the compound that's doing it. So they don't want any wood lovers getting into the pipeline. And in Oregon, it grows everywhere. So you can find some of the wood lovers out there. You can find Azuricens is one of them. What else makes this? I think some of the Liberty Caps can potentially make this. So Similianta is one that I think that can make it. So there's a handful of these other like psilocybes that don't look as if they're a cubensis per se. I think a good mycologist could probably sort them out in the field, but their concern is if those are around in Oregon, the spores in the air, maybe you get some contamination in your cubensis grow. We don't want any of the wood lovers in there. So we made a PCR test that lit up for cubensis, also lit up for wood lovers. So if there's any wood lovers there, you get a signal from that as well. And we put a third one in there for Galerina marginata and for Amanita muscara that would pick up those. So if those got it, because Galerina marginata can kill you. That one like melts your liver or something. And then Amanita, some of the Amanitas can make some toxic compounds as well. But if for any reason these guys were using the actual mushrooms and there was the mushroom DNA around, our tests would have picked that thing up and shown that, okay, this is both cubensis and Amanita, and you may have a problem here. But if it was synthetic, then there's probably no DNA there, and it would have been a bust. But
Yeah, so like 90% of the muscimol products on the market, all the gummies, all that stuff, almost all of it's synthetic. And it has the same problem you have with like Delta 8 with the isomers being not friendly and the regular base being fine. So that's at least that's my understanding is that, and in fact, who was it? Um Hamilton Morrison actually has an episode on this in the last
really?
2 or 3 months where they talked about the synthetic muscimol issue. But it sounds exactly like what was going on.
I don't know why people like that stuff, but whatever. I've never tried it, but when I read about it, I'm like, ugh, why would you want that? I guess different kicks for different folks, but it seems to be it's not exactly a hallucinogen per se, isn't it? Isn't there? There's
It's more of like a disassociative than anything
yeah.
else.
Yeah, all right. So maybe it's more of like a ketamine trip or something, but it seemed uncomfortable when I read about it.
Yeah, I mean, I've only known one person that actually like used to regularly prep it and prepare it, and that was up in Colorado. And even he like didn't do it very often. It was just kind of like, a last resort kind of thing. But you
Yeah.
have to like dry the skins of the caps for a certain period of time, and it has to decarboxylate to a certain point, and then it's psychoactive. But if you just take it raw, it's like way less psychoactive because it's not decarbed.
Oh,
So
wow.
yeah.
Oh, whole new field of chemistry there. Well, yeah. So anyway, we've sequenced these strains. We had to do that to figure out that test. The strains are now public, and a really interesting study came out of that from Alistair McTaggart. He downloaded that data, and we helped him with some of the variant calling. But then he compared it to all of the subaruginosas that he has down in Australia, because there's a different. The real question they had in Australia is when did it get there? Because humans really only figured out how to culture these mushrooms like maybe in the 70s, at least to our knowledge today. And so that seems to be fairly recent for an Australian introduction. And so they're curious, was it that, psychonauts were bringing it down to Australia and it got wild and went out and went all over the country? Or did it come at an earlier time? And his sequencing of the subaeruginosa mushrooms down there, they're still psilocybe, it's psilocybe subaeruginosa, and those ones I think do have a potential for wood lovers paralysis. And when he sequenced those, he realized that the ones in the States are really inbred and highly related to one another compared to his. So the fact that the U.S. has a market for selling spores that are asexually propagated is kind of conserving the genetic lines that we have here in the States. Um, However, his are much more outbred because they've been in the wild, and the genetic intersection of these dates back to more like the 1800s. So he thinks they got brought in by cattle. When they moved the cattle over to Australia, they must have dropped mushroom spores there, and then it was out in the wild in Australia, adapting and mutating into what it is now in the wild, and it's a much more diverse species out there, which may be helpful for trying to get some of these other tryptamines out of the plant, or out of the fungi, because there's, everyone knows there's psilocin and psilocybin, but there's also originacin, and subaeruginosa probably makes some originacin. So there's a variety of these different tryptamines in that pathway that, like CBG moving through the cannabinoid pathway and olivotolic acid moving through, these things all look like precursors that on the route to making psilocybin, they might be the tryptamines might be converted into these other baeocystin, originacin, and psilocybin and norpsilocybin. So that we have to understand, because we're only really measuring a couple of those in most markets that I'm looking at. They're just taking a look at psilocin and psilocybin and not really measuring for the other tryptamines. And if they are, they're not finding a lot of them in the U.S. Samples that are in circulation. And some of those tryptamines might have different, medical effects. They're not believed to be as strong of a 5-HT2A agonist, but they are serotonin agonists, and they hit other serotonin receptors, and they may play a role in some of this entourage effect going on in the mushrooms.
The person to ask on that is Alan Rockefeller, who actually like got in the queue to get on the show.
Oh
But
yeah,
he's
we've had some
doing
back and forth with
crazy.
him. Yeah, he's been helpful. Occasionally, we were at one point we're trying to find some Gallerina marginata in the wild because we wanted to work with it on our tests, but it's hard. You can't get them onto the biobank. We found a bunch of other Gallerinas, but the marginata one is I've not found a biobank that can supply it. So he's been out looking for that so we can just get some more validation on the test that we have. But he's really good at finding those things. He finds all types of novel ones. So he's the guy to go to help speciate these things.
Oh, for sure. And he's going to be on the show here. We've been playing tag on getting him on, and with his travels, it's not easy, but he will be on the show here in some point in the next few episodes for sure.
Yeah,
So,
I think if you go to Psilocidia, let me see. I think we have everything up there classified with a Rockefeller primer as well. So we pull that out because there is a group of mycologists that run around and do like a short Sanger sequence read on these things to try to classify them. And let me see. We had some stuff coming from Good Cap. Let me see if we have the. Oh yeah, there's an ITS sequence here that we extract out of it. This, I believe, is the ITS region that Alan is using to classify his samples. So we also have the microbiomes of these things public. If you go to Psilocidia and comb through there, you'll see some of these plots of the microbiome. So we can get a gauge of like what other bacteria are co-inhabiting these things, and if there are any variations in the psilocybin synthesis pathway. So PSID, PSIH, PSIK, and PSIM. Those are sort of the four main genes. One's a kinase, one's a methylation gene. They're all important on putting different functional groups on the tryptamine throughout synthesis.
that's so cool. Yeah, I didn't realize you had the microbiome for the mushrooms as well. That's really great.
Yeah, it's a funny thing. I think in some states that were like trying to implement this, they put in total yeast and mold testing on this stuff, and we had to like call them up, being like, "What are you doing?" The thing's a
It's a
fungi.
mold.
Everything's
It's a
gonna
fungi.
fail. So
It's 100% fungi.
yeah, we have another Oregon moment where the whole market crashes because we don't put in the right test. But
That's hilarious. Oh man.
yeah.
Well, I don't want to tie up your whole evening. I really appreciate you coming on. Is there anything else you wanted to kind of mention here before we start to wrap up?
Yeah, no, just keep an eye on CanMed. I think this year's going to be a banger. It'll be a little more affordable, and it's going to be June. I think it's like maybe June 10th, June 12th. down there, so you'll get some good beach days down there, and we should have a great lineup this year. So more of this will be presented down there, I'm sure, by then. And if you want to follow any of my other controversies in the COVID space, you can probably find me on Twitter. And Substack's a good place to go. That's where I post a lot of this stuff. Is on my Substack, which is nepentolactone. That's the compound in catnip. So I'm trying to force everyone to learn that compound's name, so I've named it that.
That's really funny. There we go. Pull your thing up. Oh, there we go. There we go. Now we can show everybody. So this is his Substack as well. You guys can check that out.
That's anandamide, and that has only one post on it. But if you go to
I'm sorry.
That's just one preprint I put up, and this is when I was first learning Substack, and I didn't realize I made 2 accounts.
Let's see if I can find it here.
But the other one has a long list of this hop latent viroid stuff's up there. And recently I've been posting on what I'm finding on all this state by state data. And please, read if I've made any errors, tell me. I'm digging into this data as rapidly as everybody else, so I'm sure there's going to be some mistakes. But I have a case study on there for like what happened in Oregon, what went wrong, what can we learn from it. And I just dug through Michigan. I'm right now going through Hawaii's data. Hawaii's data is really weird in that they run. There's one lab there that runs one platform, and they have an astronomically low Aspergillus failure rate, making me think there could be a detection issue there. Maryland has some other weird stuff going on in it, where they were moving around the TYM limits, and there's some lab shopping as a result of that. Let me see. I think if you just Google nepentolactone Substack, you'll find it. That's it. That'll get you there. And you'll see probably 50 posts there. But a lot of that is also some COVID stuff that I got dragged into over the years, because I was a little annoyed at how the whole COVID system implemented PCR. It put in a really sloppy PCR process that flagged way too many people, and they weren't even doing what we're doing in cannabis. Like in cannabis, we pay close attention to live dead PCR. Like, is it living? So we have an enrichment step in a lot of these things to make sure we're not measuring dead DNA. We're only measuring living shit, so that when people radiate things, we have we're not measuring the dead DNA. We're measuring something that's viable. They didn't do that in COVID. And so they're flagging all these people who had the virus like a month ago or 2 months ago. And that just destroyed herd immunity, because you were taking all the people who are immune and ripping them out of the population. So that was a bit annoying. So I went on a bit of a tirade on trying to bring some light onto the PCR shenanigans going on in COVID. And that led to a lot of other vaccine investigations. And yeah, that's a whole nother talk.
Well, I appreciate you coming on. We got your finally got it spelled right there and on the screen for you guys that want to follow him. And you can also find him on Instagram. I follow him there quite often. It's Twitter.
I don't post as much on Instagram because I can't. The links are always a pain in the ass, so I tend to hit Twitter first, and then if there's something good there, I'll move it over to Instagram. That can, I'll throw a link up there. Maybe it'll get bounced into Facebook, but yeah, they weren't very friendly to me over the pandemic, so I kind of lost attention span for them.
I feel you.
Yeah, that was Zuckerberg. He's coming to apologize now. He's got a lot more of that to do.
Oh yeah, I think a lot of people do.
Yeah.
All righty. Well, thank you so much for your time. It was really great having you on again. It's always fun to hear about your latest work and research. You're really kind of cutting edge in a lot of parts of the space, and I look forward to working with you on some of the other projects we're working on behind the scenes that'll come to fruition. I'm super stoked.
Yes. All right. Well, thank you for the time. Appreciate it. And I'm glad. I always learn something. I learned musk mole is now synthesized. I was wrong about that.
Well, one of the other ones, and I know I mentioned it before, but with the mosaic virus, when I was working at this one facility in the South, they had 4 different rooms set up with 4 different lighting companies. And one of the lighting companies, all the same clones, same mothers, like each row was exactly the same clones, so that they were as even as possible to judge yield. and the one particular light spectrum was causing, and it could have been HLVD. I don't know what the causes was causing the chlorosis. I'm assuming it was mosaic virus because that's what it visually looked like. But it caused just that one room to full blown express that virus, completely, whereas the other rooms were completely unnoticeable. And you could see the buds were much smaller in that row, the ones that were all messed up compared to the other three rooms. But it was like just the light spectrum was the only variable. The newts were the same, the air conditioning was the same. It was just the light spectrum. And I always thought of it as like a way that you could like screen a bunch of genetics, right? So you do your pheno hunt, you can just put a bunch of clones underneath this light and immediately screen like 1,000 clones. You know what I mean?
Oh God, there's so much
For testing.
Mark has sent me a link to somebody who's doing something on the biocontrol front where they have something that is helping with yield, but they think may also have an impact on HLBD. And I'm trying to get him in touch with Samir and a few other people because to me, I'm like, it makes total sense. There's got to be a way to deal with this.
But if you could force trigger it to be able to find it that way, it might help like do large assays
it's
cheaper.
Like, yeah. People sometimes do that for finding males too, right? Flip the lights early and see if they one way of screening for sex, I suppose.
Yeah, well, that's usually why we do it. We grow them up big enough for one clone, take it, especially if we're doing a lot of plants, and you don't have them tested genetically. It's kind of the old school way.
Yeah, and it's also got me thinking about the rooting hormones are one area that I'm curious to what degree those are triggering it, because we do have some links in the HLVD pathway to some of those hormones. I don't know whether it's which way is regulating it though. We've got to do more work on that. But it got me thinking that we do tend to see this more in clone houses where they're dousing these things with rooting hormone, and maybe we got to think differently about that.
I think a large part of that is that people used to sterilize with rubbing alcohol because that was the standard for so long. And we know that doesn't work for HLBD. So you have to do that like 10% bleach solution or 15% bleach, whatever it is. I forget the exact number.
Yeah.
But I know how much to fill up a party cup. That's all to not fuck it up. But you put that much bleach in there, and that's what actually kills it. But I think that the facilities that are doing that, I bet you are having less transfer from, because I've even gone to facilities, especially in Oklahoma, where you have very obvious mosaic virus and like one or two of the moms. And then by the time you make it to the flower room, it's in like half the strains because they're not cleaning the tools right,
God, yeah. Yeah. And some of these things you can't, there's the fact that the thing that scared me about some of today's data we got was that we didn't see it in veg in the roots, which is like, damn it, if this thing hides and it can show up when you flip the lights on, then it really changes the way you got to screen for things, right? You're gonna have to do testing and flowering. You can't have 100% clean bill of health if there's things that can hide this on you in veg. And that's I think a lot of people were hoping, particularly the clone houses, they'd be like, all right, they can tell you it's clean at that stage. You bring it on, it's clean maybe the first 2 or 3 weeks in veg, you flip the lights, and then you have to start questioning, okay, who introduced this? Like, did we introduce it at our grow, or is this something that was hidden from a clone house? And you can't necessarily blame the clone house because we're having a hard time finding it in some of these plants in veg state. And then we're not a huge, the people that we're working with on this, they got boutique numbers of plants. This isn't like a huge house where there's scissors getting shared between plants and everything. They're just handling these things. So they couldn't, and they're not outdoors, so there aren't bugs moving this stuff around, and they're putting them in isolated buckets. So this thing is in some plants it can hide from you in veg state, which means it just changes the way we were thinking. Before we thought we could screen the hell out of this stuff in veg, and now we're finding plants that can evade that, and you only find it in flower, and it's not introduced by anything but the injection we gave it. That I don't know. We have to get our head around what to do there. That was something we just found this week, so it's one of those things where we're like, oh shit, this is a light bulb moment on how we do screening.
Yeah, no, that's super important too, because like you were saying, you could buy stuff from somebody and it totally tests out clean, and then when you flip it, it doesn't. It suddenly is expressing, and you're having problems. So that's something I learned today talking to you about this. So
Yeah, yeah, that's not something we were thinking was on the list a year ago. That's something that just it's happened to us already, and on when we injected this particular Jamaican line cross, and it hid from us and we didn't cut anything short in veg. We had that thing in veg since like I think it's like May, so it's like what is that, 5 months?
Yeah, it's a long veg time. You
Four
should have
four
seen
months.
some expression. Yeah.
Yeah, it's 4 months. And because it was in August. Yeah, it was in August when we had. So it should have like it was late veg, and it never showed. We tested it every week, and then bang, you flip it, it's there. Now, granted, we didn't have it infected that whole time, but we did have infected for like a full month, and it wasn't around. We couldn't detect it anywhere after we injected the damn. I mean, we didn't the day we if the day we injected it, we could see it, but then we walked away for 2 weeks, being like, we got to wait for it. That's a going thinking, is you need to wait for 2 weeks for it to move around if you find it everywhere else. And we didn't see it anywhere after that. So we were about to re-inject it, and then we said, no, wait, flip it, and let's see what happens. And then bang, it showed up in the roots, but not in the canopy.
Yeah, no, that's super interesting, and certainly will help us eventually figure this nut out, because that's game changing information.
Yeah. Well, sorry to be the bearer of bad news if it's bad news, but the silver lining is I think breeding our way out of this is going to require CT scores. Like you want to be able to quantitatively measure how much you have and not just take a blind, cut everything down. Because I think the plants that like keep it suppressed at low copy number in the canopy, or just don't even only keep it in the roots, are the things we got to start working with. And breeding those may get us we know at least in one cross that has transferred. I don't know what gene it is, but when we crossed Jamaican line with Mandarin cookies, the offspring had it, and then that it keeps it in the roots. I don't have a Punnett square to tell you whether it's recessive or dominant, but we'll get there. We can do that. We've got more seeds, so it is something heritable, and that means there's a way to breed out of this.
And that's how the hops community solved it, correct? Or like
Yeah,
mostly
yeah,
solved it.
they never got rid of it, and they've been dealing with it for decades. So I don't think we're going to come up with a magic wand that gets rid of it either. But we got to start profiling the plants with a little bit more information than just presence absence. We got to look at like what's the viroid load and where, and that's going to probably teach us what to do here.
Awesome. Well, I don't want to tie up your whole night. I'll let you go.
Cool.
I've already taken up 2 hours of your time. I really appreciate you coming on and sharing all this awesome knowledge with us today. Thank you so much.
Yeah, thanks, Steve. Good seeing you. Cheers.
Yeah, nice to see you as well. Take care. Well, that was an awesome episode. And if you guys are looking for additional education, be sure to check out medicinalgenomics.com, as well as the Instagram and Twitter and all of his other awesome projects. And trying to pull up, sorry, I'm having an issue here. Pull this up. If you guys are looking for aquaponics education, before we wrap up, I have put out a new aquaponic master course. This is almost twice the length of the original cannabis class, with a ton of new information. The first block of the class is now available. I'm just editing up the rest. The rest will be up here in the next month or so. But you can sign up now and check that out. We go through everything from design work to build outs to all the different aspects of fish. We cover over how to grow over 100 different types of crops, all the different ways to set up the root system depending on what type of crop you're growing, fish disease, fish species. Over 40 different fish species are covered in this course, as well as almost 200 slides just on pest control alone. And all the different bugs that you're going to run into, all the biocontrols, the beneficial insects, release rates, application rates. You'll also be the first to access the new aquaponic assistant AI that I've been working on that does pest control and aquaponic nutrient dosing. So you can check that out over at aquaponicmasterclass.com. You can sign up there and get instant access to the course. And it's the kind of like I don't know, a combination of a bunch of my different classes plus about a third a new data and new applications with a bunch of refinements and a lot more exact data on dosing and release, release rates, treatment rates, all the microbial recipes, including a bunch that we've never talked about on the show, and a whole bunch more. So check that out at aquaponicmasterclass.com. We also have a bunch of other courses available over there as well on Teachable, and you can check those out on as well in the description of this podcast and whatever platform you're listening to. All right, thanks a lot for watching, everybody. We'll be back again Thursday or Friday. A little bit of a unsure on which day we're going to do the next podcast next, but we should be back to once a week now. I have an office again and a studio again and everything. So, yeah. And if you're in NorCal, hit me up. We got some cool projects going on. Alrighty, guys. Thanks a lot. Like and follow, subscribe.